I will talk today about the cutting-edge technology of bio 3D printing. And my agenda today will be new high-tech and 3D bioprinting process, video illustration, real experience, published work in GI and different subspecialties, and summary. And what is 3D bioprinting? 3D bioprinting is the method of printing biomedical structure with the use of viable cells, biological molecules, and biomaterials. And Nakama et al. 2013 developed a robotic bio 3D printer system called Regenova that blazed spheroids, which is aggregates of cells in the fine needle arrays according to the pre-designed 3D data to facilitate fabrication of 3D cellular structures. And this is the shape of the needle array. It is of different sizes and it is appointed as according to the tissue to be manufactured. And regarding the process, step one is spheroid or cell aggregate preparation. The culture of enough cells to form a spheroid by aggregating the tens of thousands of the desired cells. And then stacking or shoving of these spheroids into the needle arrays to form the tissue, the structure that we want. And after that, let these spheroids fuse together and we will get the desired tissue. After that, we remove this needle array and we will have a 100% cellular bio 3D printed structure. And here, as you see here, this is the shape of the structure after we have bioprinted. And then we bought this tissue in the bioreactor, which is like incubator that provide the tissue or that provide this structure with oxygen and nutrients for completion of the process of maturation. And here is a video that illustrates the whole process. This is the machine that is used in the bioprinting system. And this is the software where we can design the tissue that we want. For example, here, this is a tubular structure. It can be esophagus, trachea, or a blood vessel. And here, all these are automated process. And then there's the process of 3D printing. Here, we can select the spheroid, take by automated syringe by process of aspiration. And then we bought this spheroid in one of the needle, like that. We shove or stack this spheroid into the needle, and we will repeat the process again and again. And the amazing thing here, we can modulate the structure according to what we want. And let these spheroids fuse together to form the tubular structure according to the design in the software. And after that, we will remove this needle array. And we have a bio 3D printed 100% cellular structure. And I was lucky to have witnessed this technology in my preceptorship in the University of Nagasaki 2016. There was a project in collaboration with the GI and the cardiothoracic department about this technology. And we have printed this structure. You see, it is elastic, but it is real, 100% cellular structure. And actually, we transplanted it in a rat, in the trachea of a rat. And here is the result. You see the difference. This is a yellowish color before transplantation, and this is a pinkish color after transplantation. And the amazing thing about this technology is the tissue, the structure acquires a micro vessel-like network that permits blood flowing into the tissue for proper integration into the adjacent tissues. And fortunately, with no rejection. And regarding the published work, this process already successfully fabricated blood vessel, beating heart, and the implantable grafts like trachea and urethra. This technology is also capable of making a small tissue piece of in vitro research which mimic in vivo. Actually, this process has succeeded to fabricate a liver tissues that are able to secrete bile and metabolize drugs. And also, these processes have succeeded to bioprint esophageal tissue, pancreas, and also intestinal tissue. And in a nutshell, scaffold-free tissue engineered structure could be a promising solution to create tube substitutes for clinical transplantation and to optimize culture conditions and cell combination used to generate scaffold-free structures by a bio-three-dimensional printing system, notably long-term culturing with proper medium flow promoting extracellular production sufficient to increase the tensile strength of scaffold-free structure. And these data provide basic information for preparing a structure for implantation in animals. Tissue engineered scaffold-free tubular structure with a micro vessel-like network could be a significant step towards the clinical application of bioengineered organs. And thank you.